This is my interview with James P. Allison, PhD, who is the Director of Immunology at the MD Anderson Cancer Center in Houston, Texas. As you may know, Dr. Allison won the 2018 Nobel Prize for his discovery of immune checkpoint inhibitor drugs, specifically Yervoy. This drug has been very successful in treating melanoma, and in combination with other immune checkpoint drugs like Opdivo and Keytruda has really constituted a kind of revolution in the field of the immune approach to cancer.
I've been writing about cancer and specifically about cancer immunology for 45 years. And I started as science writer at Memorial Sloan Kettering. So that's how I came to know about this. And I don't want to make this about me, but just to give you an introduction, that I wrote the first article for the general public about tumor necrosis factor. So this is how I came to know Lloyd Old and Helen Coley and all those people. So I wanted to start by asking you about them and about the earlier history of immunotherapy, because it seems like, in a way, your work is a culmination, or certainly a new advance in terms of the practical application of immunotherapy. But you won the William B. Coley Award. I believe that your wife also won the William B. Coley Award, and you were the chairman of the Scientific Board of Cancer Research Institute. And I think you and Lloyd Old were actually over the years the only chairman of that board,
Yeah.
right? So
Right.
Could you talk a little bit about Coley and about Helen Coley and Lloyd Old, how you see that development and how that fed into, if it did, into your own work?
Well, of course, Coley was noted for his work as a surgeon, noticing that when patients got infections, they tended to not head and neck cancer that he did surgery on, they tended to recur less often than those who didn't. Anyway, I think that I'm not sure that he thought that was really immune related or was just the property of some bacterial toxins, because he called them toxins. But I think that the modern view is that he probably it was properties associated with the toll-like receptor agonist that bacteria have that alert the immune system, that something's going on, the innate immune system. I really wasn't aware of that work until I, became associated with Lloyd, and I met some of the family, but I'm not sure I ever met his daughter, I think. But I have been associated with the Cancer Research Institute for many years.
Was Helen still around when you became associated?
No, I don't think so. At least I never met her. I know some of the other members of the family, but not her.
And what about Lloyd Old? I think I heard you speak at his memorial service
Yes.
10 years ago, and I remember some astonishing statements that were made at that service in New York that Jill invited me and many other people to, of course. Where do you see Lloyd Old's role in the larger picture of the history of cancer immunotherapy?
Well, I think that some people call him a grandfather or godfather or whatever. He certainly was involved in some of the early mouse studies, actually demonstrating that there were antigens and it was possible to get tumors rejected, and keeping the idea that one could use it. The first time that I'm aware of that the word that somebody actually proposed that the immune system could be used to treat cancer was Paul Ehrlich in 1906. He talked about antibodies could be the magic bullet, but he was thinking about antibodies to the tumor cells themselves. Never really took it anywhere, but the idea had been around. Of course, in the 60s, when sort of the idea of immune surveillance came to the fore, And people began to think that, first of all, that's why we have an immune system, which I think is not true. I mean, I think that's demonstrably not correct. On the other hand, we have an immune system, and it's demonstrably true that it does, have anti-tumor effects, or can be tricked to. I mean, I think the ultimate origin is irrelevant, but there's plenty. Bob Schreiber, his work recently, also through, Lloyd support and through the CRIs, really, given that a real boost. But I credit Lloyd. I first became aware of his work in the '60s, and I'd had, I was not particular. I lost a bunch of family members to cancer when I was young. My mother when I was 10, and a couple of uncles within a few years of that, and my brother, and it goes on and on. So I was interested in cancer. I was a biochemistry major, really, and was trained in biochemistry in graduate school, but I took an immunology course, and just after T cells were discovered, and the professor was talking about them, and I just got really fascinated, because I said, it's these cells, they go all over your body and go through your tissues and through your blood and protect you against stuff. I said, how does that work? And I went to see this professor. This was in about 67 or something like that. And, he said, we don't have a clue. I don't know. And he even doubted that they existed. He said, I think they're just other cells. He was an antibody guy. He said, I just think that's some kind of macrophage-like thing that's picked up an antibody. So he didn't eat that, but I decided at that point that's what I really wanted to work on. And then, of course, because of my family history, I always thought about, cancer. Maybe we could do something. And I went to the library. At that time, we actually had journals, books and stuff, and read a lot about Lloyd's work, and then about T cells, the T cell receptor, and it was. I just decided, that first of all, I wanted to unravel the secrets of T cells, and as a second part of that, I figured when we figured that out, that maybe we could do something about cancer. And to tell you the truth, I was a little skeptical of a lot of the work that was going on, because everybody with TNF and with Coley's toxins and with the tumor vaccines, what people were trying to do was alert the immune system, stimulate it, give a positive signal, and get it to doing stuff. And that is, they're aware. I mean, that stuff in human trials at least was not successful. None of those things really, including Steve Rosenberg's work with T cell growth factor. This was known then, IL-2 now, but that had a response rate of somewhere around 5 or 6% in melanoma, which is slightly higher than the spontaneous remission rate, but not that much. It also made people very, very sick. Finally, there's no evidence really that it does work through T cells. I mean, there's just no direct evidence of that, ironically enough. Anyway, but I was there, and I was, every now and then I do a little tumor immunology, but really I was just trying to understand how the immune system worked. And then we had autoimmunity models on one side and cancer on the other. And so we were simultaneously in animal models trying to make autoimmunity really bad and then try to use that.
But it was basically the discovery of the CTLA-4 that then led you to develop ipilimumab as a treatment?
Yeah, but before that, I got interested because, I was the first to work out the protein structure of the antigen receptor in '82. And, then it became clear that there were initial signals needed, co-stimulation. And so we showed that, CD28 was that other molecule. Then CTLA-4 came along mainly because it had been cloned by another group in France. And when we cloned CD28 and did homology searches, which were pretty crude back then. But anyway, the CD28 showed that there was a sequence comparison showed that CTLA-4 was very much like CD28, and so that led me and a whole bunch of other people to start working on CTLA-4. And so the first notion that was published by Peter Lindsley and Jeff Ledbetter and others was that it was another CD28 molecule. You know, it was another gas pedal.
So basically, if I'm understanding correctly, what the ipilimumab and some of these other drugs do is essentially release the emergency brake on the immune system, right?
Yep.
By getting between the cancer cell and the immune cell, to put it in very simple layperson's terms,
Yeah.
right?
Yeah. Essentially, although it's a little more complicated than that. But before we got to that, you know, I had the idea that maybe one of the reasons that vaccines hadn't worked very well, and that you know, a lot of other things had not worked, is that tumors didn't provide that co-stimulatory signal.
You said in your Nobel Prize lecture that basically there have to be certain dead cancer cells in the system in order for these drugs to really work. So I think what you were aiming at, if I interpret it correctly, was the idea that you do need some other treatment, something that's going to kill the cancer cells, but wouldn't, like using typically high-dose chemotherapy or radiation therapy, wouldn't that also diminish the number of lymphocytes that the person has to work with?
Depends on how you do it, but yeah, I mean that is one of the problems, is that really high-dose radiation, high-dose chemotherapy, a lot of them, you know, do obviously immunosuppress people. But on the other hand, there've been a lot of studies recently that show that a lot of the chemotherapies don't work all that well in animals that don't have immune systems. You know, in other words, that there is an immune component to even the chemotherapy. But what puzzled me, you know, was why, because there were all these ideas, oh, we have cancer all the time and the immune system exists. You know, that whole idea was that the immune system is designed to hit cancer. And yet, when we started working on co-stimulation, we really quickly realized that there's a 3-cell assay that you could do, you know, where you had one cell with an antigen on it, and then another cell that didn't have antigen but had what turned out to be a B7 molecule, which binds to CD28. And you could, you know, have a T cell there, and with those signals on separate cells, sometimes you could get T cells activated. But without that second, and then dendritic cells can give all the signals. But what we did was we took the ligands for CD28, these B7 molecules, and put them into tumor cells. They got rejected immediately. So that proved to us that it wasn't lack of antigens. It was because the immune system, the tumor cells were invisible to the immune system. Those antigens were invisible to the immune system because they couldn't give that second signal. But when tumor cells die, you know, it alerts the innate immune system. The dendritic cells come in and phagocytize the antigens from the dying tumor cells, put them on their surface in the context of those B7 molecules, and that's when the immune response starts.
So would it make sense to give a dendritic cell vaccine in conjunction with the immune checkpoint inhibitors?
Yeah, so people did that quite a bit, and that has never worked particularly well either. I think because what we realized, you know, when CTLA-4 came along, is that, you know, a T cell that's just going around, it can bump into an antigen-specific, you know, a T cell that would have a receptor specific for something in the tumor. Without that second signal, there's no response. You know, but once you get it started through this cross-priming, when the T cell gets the signals, what happens is there's 2 parallel pathways that are turned on. When you get CD28 and T cell receptor, one of them is just a cascade of signals that tells the cells to start dividing really quick. So you get clonal expansion, and then, you know, they differentiate and start to acquire effector function. But the other thing is you turn on the CTLA-4 gene, because that CTLA-4 is not expressed in a resting T cell. So that you turn on the CTLA-4 gene, and that starts this negative process that is going to terminate the ability of CD28 to co-stimulate, because you have to stop that expansion.
Would you say, broadly speaking, that the phase that we're in is one in which cancer is being redefined as an immunological disorder? Because you said in your lecture that basically responses that are caused by conventional chemotherapy drugs are basically an extension of life by usually a few months, but don't really lead to long-term survival, right? But when you use the immune checkpoint inhibitors, or broadly speaking, immunotherapy, you're seeing something entirely different that had not been seen for
many, in some years. Patients, yeah.
So you said in the lecture, I think 20%, roughly 21% of the patients who were on the Yervoy or ipilimumab were having, if they made it to 3 years, basically they were having long-term survival, that there's basically no fall off. So in effect, what you're showing is, practically speaking, a cure, if you define cure as living with your disease or not dying of the disease. Right. Are those people, that 20%, have they had complete responses, or is there still tumor left in their left in their system?
In most of them, in fact, in all of them that I know of, there's no detectable tumor anymore. Now, that's not to say, of course, that there's not some little islands of, small micrometastases of tumor somewhere, and that's the problem, with using the word cure, just to depends on who's defining it. But I've given talks, particularly to surgeons, cancer surgeons, and I was giving one time, and this, and I said, we've got patients now from a study of 5,000 patients. There have been hundreds of thousands treated now, but about 4 years ago, there was a retrospective analysis of 5,000 patients who had been treated. And that was the one where there was 10 years follow-up, and that was the one that showed that a little over 20% are alive a decade later. And there's a woman from the phase 1 trial of IPI from 2001 that she got a single dose. She's still alive. She's almost 19 years out now without any more treatment, without any recurrence. And so, anyway, this surgeon stood up and said, Jim, you just can't use the word cure, because cure means there's not a single tumor cell live, in the body. And we see spots on CAT scans. So you can't say that they're tumor-free. And I said, well, I'm aware of many, many, many cases like that. But if you biopsy the tissue where that spot is, when it's been done, there's never any cancer in it. It's always a granuloma. It's a result, it's like a crater in the battlefield, it's scar tissue where the fight was going on. And so it's a semantic question. But that doesn't mean there's not a few tumor cells. And I said, but remember, you're treating a patient, not a CAT scan. And I feel pretty strongly about, at 10 years with no recurrence and no necessity for additional treatment, giving people a break. And, because if you tell them you got a chronic disease and we got to keep checking because it can come back, I know patients, and that's really tough on them when they keep having to go back every year, and and there's this syndrome associated with this fear every year. And I think, and I know one woman, her doctors told her, don't have babies. You should not. Of course, chemo is not an issue, because with chemo you can't have children because all the germ cells are destroyed by the chemo. And with radiation too, depending on how it was given. But what the concern with ipilimumab is that women who get pregnant, there's a phase of the pregnancy where they're pretty strongly immunosuppressed. It's just naturally, by hormones and stuff, and partially to protect the developing fetus, from attack by the maternal immune system against paternally encoded genes. But anyway, and so, they were worried with the first women. This woman I know was 22 years old when she was diagnosed. She had brain mets, liver mets, lung mets. She had everywhere. And, she was hospice bound when she got ipi. Her tumors went away. And, that was 14 years ago. So she's one of the early people in it. But a doctor said, you really should think about not having babies. And so for several years she did that. And finally, after a while, she just said, to hell with melanoma. I'm going to get her down. I'm going to have kids. I'm going to live my life. And she did. She just, said, I'm having kids. So she's had 2 kids, and everybody's fine. One of them's 10 now, I think, and the other one's 7 or something like that. I saw them recently. Which kind of begs the question, but back to the thing is that the real issue, I think that we can say the word cure. I mean, I think, and not lightly, because there are people who, relapse early on, but, it's about 3 years. But now with the combination of anti-CTLA-4 with ipi plus nivolumab, a PD1 drug, about somewhere close to 60% of patients have complete responses.
Of melanoma patients.
Of melanoma against, stage 4 melanoma.
Which is astonishing, never heard before, 60% of people advanced. Usually.
And these patients, those patients have only been tracked for about 5 years now, because that's what the trial was. But it still is between 55 and 60% at 5 years, so it's likely that that's going to go out.
But why? Two questions. One of them is, why is there such? You yourself said in your Nobel lecture that that the responses are very different in terms of the different kinds of cancer. You would like to extend this to everything because you, as you eloquently said, the immune system goes everywhere. The immune system is the basic antagonist of the cancer cell. And and this is not chemo. This is because these drugs don't kill cancer cells, which is a concept very difficult for most people to wrap their heads around. And yet we see such dramatically different responses to the immune checkpoint drugs. So could you address that?
Cancers, you mean?
Based upon the anatomical location of the cancer.
Well, it turns out it's really not anatomical. It's what caused the cancer. Melanoma is caused by ultraviolet radiation, from sunburns. That's pretty clear. And what's also clear is that melanoma has a really high, mutational burden. Team B, it's called tumor mutational burden, relative to other cancers. In the hundreds to thousands of mutations per cell. And now we know from some work that we've done, and I shouldn't take credit for it, but Bob Schreiber largely, in mouse models and any number of people that have followed him into human tumors, that the immune system in cancer tends to be preoccupied with neoantigens, that arise from mutations in peptides that can be presented by MHC. And so, melanoma, if you have thousands of those, you're a lot more likely to have some that are going to be really good targets for the immune system.
So the immune system is going after abnormal things in the body.
The thing that's cool about that is that they're also, anyway, melanoma responds to monotherapy, of course, with ipi, and then also even better when you do combined therapy. The same is true of lung cancer, which of course is caused in large case by carcinogens from smoking, as is head and neck cancer, throat cancer, bladder cancer, some kinds of stomach cancer. All of those have, again, high tumor mutational burden, and all of those respond very well to immunotherapy.
All the things that we
All
can
of them.
we can identify carcinogens as having caused, right? Those are all tumors that are associated with powerful carcinogens. Repeated insult, repeated
Well,
insult.
it's probably with chemical smoking. It's probably over a long period of time. This is something that's of interest. It's kind of arcane, but probably sunburns. You would get you got a really bad sunburn. You probably got hundreds of mutations all at once. And then you do it again and you get hundreds. And so. And that can be tracked by people building phylogenetic trees from the mutations, branch mutations and root mutations and all this stuff. You can construct, evolutionary trees of the cancers by their mutations. But the real thing that's about that's interesting is that those cancers were almost impossible to cure with chemotherapy, and particularly the targeted agents, because you can identify the driver mutations. That's what cancer biology, that's what the whole war on cancer was about, making an inhibitor to BRAF, which is mutated in 60% of melanomas. And you can have a drug that'll, kill that pathway, you get a mutation, it activates BRAF, turns on the whole MAP kinase paper, tells that pathway, tells that cell, keep growing, don't stop. It's really pretty easy to inhibit that enzyme. And then the tumor cells all die, from what you can tell. This all started happening about 8 years ago. But then it comes back. And that's because with so many mutations, there are other drivers that are there that are in a tiny fraction of the cells. Or there's new suppressor genes or something. For BRAF alone, it's been shown that there are 13 ways, at least, that a tumor cell has of getting around, blocking that mutation and keeping going, and that's you cannot, because of that genomic instability and the high rate of mutations, you can't treat them by targeting those mutations one at a time. It always comes back, but because they have those mutations, they're sitting ducks for the immune system. It's an interesting thing because that very complexity that makes them so hard to treat by conventional makes them sitting ducks for immunotherapy.
So you kind of flipped oncology on its head. The things that were the hardest to treat now are the things that are most effectively treated.
Yeah, that's the way it seems.
And what about the gut microbiome? There have been so much talk about this, and I know that the Parker Institute, with which you are affiliated, has come out with this astonishing report about the presence or absence of microbiome or of variety of probiotics, if you will. But it seemed to be sort of counterintuitive, because what they were saying is that if you have had antibiotics recently, you would have a diminished immune response. But they also said that if you were taking probiotics, it also would be. Can you explain that?
Yeah, so that work actually, there's 3 groups that have done it. One in Paris, one in Chicago, and the one that the investigators working with the Parker Institute is Jennifer Wargo, who's here, and I've been collaborating with her for some time on that work. So it's very clear that what you just said is correct. What she showed was the more diverse your microbiome is, the better these therapies work. So in other words, if you narrow it to some bacteria, so that implies there's good ones and bad ones, and so kinds of bacteria. So now, of course, that's what everybody's trying to do, is find the good ones, make probiotics. But what happens when you give the right probiotic? What happens when you give probiotics that are out there now is that the stuff you put in very often dominates the microbiome of the people who are taking them, and it becomes less diverse. And that's probably the basis of why, those things are not. We knew the right bugs, but, maybe you could do that.
Do you think there would be some benefit, though, in terms of eating fermented foods? in order to get a diversity of microbiomes?
Uh, I'm not sure. I mean, maybe. Certainly, just to say it's diverse does not tell you everything you need. But there is something going on. When I first heard rumors about this, right after I moved to Houston about 8 years ago, I just had a technician in the lab work with our standard system, mouse tumor. We used melanoma. We also used a colorectal carcinoma. But anyway, put it in mice, and we could cure those with our antibody that we made to CTLA-4, the original antibody there. We could cure those mice. But if we just got all the mice, got a bunch of mice together from Jackson Labs or wherever, randomized them, and then gave half of them antibiotics, to sterilize them as best we could, one group, and then the other group didn't do anything to. They had all been housed together. And then we gave them tumors and treated with anti-CTLA-4. It did not work in the ones that had gotten antibiotics. I mean, we could see that in the mice. It was totally ineffective.
The drug was ineffective.
Yeah, and so subsequently, we weren't the first to do that. I didn't know about it when we did it, but 3 or 4 other groups had done that as well and published it. And then people then started looking at the clinic, and sure enough, people that got infections, have poor outcomes generally. And so doctors are warned now, don't give antibiotics unless you have to. Don't give them if somebody gets cold, or something like that. Just stay away from them until, of course, if somebody gets really sick, you have to give them.
Have you heard about these pilot study about doing fecal implants for people? What do you think about that? I mean, it's being used for autism now. People in Israel, I think, are experimenting with this for cancer.
Yeah,
it seems as if you can transfer the susceptibility to or the sensitivity to the immune checkpoint drugs if they get an implant from a person who's already responded to one of these drugs. Do you believe that, or do you think that's promising?
Yeah, Jennifer has some results like that, as does Laurence Sitvogel in Paris. We're working with Jennifer now. We share a graduate student, and so Jennifer's treated hundreds of patients now and gotten specimens from them, stool specimens from them. And so where my lab comes into that work is we're taking our animal models and giving them those fecal transplants from the humans, that responded well, and then giving them the mouse tumors and seeing, we're just getting started, so I don't know if it's going to work. But what we hope to do then is use, I think it's going to have something to do with toll-like receptors or something in the bacteria, not the bacteria themselves, but some product. But we hope by doing this work in mice, we can use, genetically modified mice that lack, one of the pathways of toll-like receptors or whatever to really narrow it down and figure out what the mechanism is. But so, she certainly got there. What I know that's already being done, and it's also being done here, is that, my wife's you already mentioned she's a GU oncologist, and in the GU clinic, one of her patients, but there was a woman who was, actually been several now, who was being treated, I think with the combination of anti-CTLA-4 and anti-PD1, but anyway, developed colitis, a really bad colitis, and so that's a fairly common adverse event associated with these drugs. And normally what happens is it occurs kind of late, after several treatments, and you can treat it with systemic steroids, and are things that block TNF, activity, and still get the anti-tumor effect, but cure the colitis, and then you wean the patients off of the immunosuppressive drugs, and they're fine. It does come back. So it's not autoimmunity. It's just temporarily imbalanced. Undoubtedly, there's T cells involved in it, but it's not really against self-antigens, most likely. But anyway, so they had a woman in the clinic here, GU clinic, that they couldn't touch it. They tried, steroids didn't stop the colitis. They tried some of the anti-TNF, other stuff, just couldn't touch it. So they just did the experiment, just got fecal samples from somebody who was normal, and gave them to this woman. Within 48 hours, everything was fixed. She was back to normal again. And so now there's been a lot of anecdotal stuff, but now there's a formal trial, started to really test that. And so,
I remember a study that it was from MD Anderson, but I think you were senior author on it, where you analyzed responses based upon the dose of the drug, the immune checkpoint drugs that were given. And in terms of survival, I don't think there was much difference between the high dose and the low dose. Am I summarizing that correctly?
Yeah, I was an author of that. I didn't do much on it, but yeah, I mean, I think that we're overdosing. I think that we're probably overdosing patients now, particularly with some stuff that we've learned recently about the mechanisms of how it works. We've been using a lot of single cell analytical techniques to look at how these drugs work, and I think in some cases we're overtreating.
So, what would you think about a program where they used the lowest effective dose in combination? Wouldn't it? I mean, because the toxicity of combining the anti-CTLA-4 drugs and the anti-PD1 or PDL1 drugs, it's considerable. Or even with the high dose of ipilimumab in the original Hodai study, if I'm not mistaken, as many people died of the treatment, or at least the combination of one or the other drugs that were given in the treatment, as were put in complete remission.
Yeah, some people died in the early trials because of ipilimumab, and the reason was because we never saw any adverse events in mice except some vitiligo and melanoma models here, which was expected because the T cells are attacking normal melanocyte antigens. But we never saw anything else, no weight loss, no anything. And then in monkeys too, of course, the ipilimumab, we made sure that it tested it and showed that it bound to cynomolgus monkey CTLA-4 just as well as humans, and then gave them doses 10 times, 100 times as high, and for 6 months straight, no adverse events whatsoever. And then it goes into people, and bam, there's stuff. And I think the reason is that the mice, are kept absolutely as clean as we can keep them. And then monkeys tend to be, even though they throw stuff around, most people have had thousands of infections, and so it's easy to, I think what's going on is just, it's complex. And you could shift the balance after a while.
Well, what I'm saying, what I was getting at was, if you give a low dose of these drugs, don't you diminish the possibility of these severe grade 3 to 5 side effects by doing that? Wouldn't that be a pathway to using them more effectively?
Yeah, but there's been at first, Mr. Mouse Squibb, kept track of adverse events. First of all, it was lethal in some patients, but very quickly, like with any drug, the physicians who used it developed algorithms for treating it. So almost nobody dies of the colitis anymore. That was what killed people early on. There's some new adverse events which are appearing now that hundreds of thousands of people have been treated. But what appears now is there is a correlation between adverse events and response. So in other words, if you have an adverse event, you're more likely to have a satisfactory outcome. Now, that's not to say that you can't ameliorate that. An example of that is a study that we just did. Actually, we meaning my wife actually did it in prostate cancer. This is an interesting story for several reasons. One is that prostate has very, very low mutational load. It's about at the level of it's colder than pancreatic cancer, for example. But yet there were responses with ipilimumab as monotherapy very early on. It went all the way to a phase 3 trial, which failed because there weren't enough to make it really different than standard of care. But and then PD1 was tried, and there were no responses whatsoever to PD1. And so BMS just shut down their whole prostate cancer program. But we talked them into, I began to have the suspicion, that, if you have a cold tumor, no immune infiltrates, checkpoint blockers aren't going to do you any good because there's no T cells there. You've got to. But anyway, we began to suspect. I won't go through all of it, but anyway, so we got that IPI might actually drive T cells in, and so we got some patients. My wife specializes in pre-surgical treatment of patients. Before a patient with localized disease that get a couple of doses before they go there to surgery. Anyway, so we had biopsies, cold as it can be, looked like pancreatic cancer. But after a couple of doses of IPI, they look like melanoma. There's T cells, macrophages, it's full of
So
stuff.
the cold hot scale, is that a measure of the number of tumor infiltrating lymphocytes that are in the cancer? So it just it's sitting there, the immune system is not reacting to it. That's what you mean by cold, right?
Yeah. You could have cells so that very often, if you look closely at samples, you can see that tumors will have T cells around them, but not inside. And
Is
they don't
that still
respond.
still hot? Is that considered hot? If you've got
No, that's considered cold.
That's still cold, but the cells are not infiltrating the tumor. That's still cold. But when you give the Yervoy or the ipilimumab in advance, it drives the T cells into the tumor, so it makes it more. I'm just paraphrasing what you're saying, but it makes it more like a very highly mutated cancer like melanoma.
Yeah. And then what happens though is what our work told us from back in the 90s is that, actually something I got off the subject of, but rather than trying to turn T cells on, what I was trying to figure is how to keep them from being turned off. If you know what I mean? That's a very different thing. Not only were we not treating the cancer, we're not trying to turn T cells on. We were trying to keep them from being turned off by the normal control mechanism. So, what we were doing was, that made it controversial because I said, first of all, we're not even treating the cancer. And secondly, we're not trying to treat it. So people said, that'll never work. But it turns out it does. But anyway, when the T cells go in after the IPI treatment into prostate cancer, and pretty sure in kidney cancer as well, they also begin to express the whole PD1 pathway at very high levels. It's a compensatory thing to try to keep them in check. And so there's PD1 and PDL1 all over the place. And the PDL1's on the tumor cells. It's on a subset of macrophages. It's on the T cells themselves.
So could you take, since to a certain degree, people are still looking at PD1, PDL1 axis as an indication for whether or not immune checkpoint drugs would work, could you...
Yeah, that's a big mistake.
Big mistake, right? So that's been sort of gone out the window. But if the people are still thinking in terms of PD1 and PDL1, you could, if I'm extrapolating correctly from what you've just said, you could pre-treat those people with Yervoy or ipilimumab, and then they would start to express PD1, PDL1. You think that?
So that's probably, so we showed our results at Bristol Myers Squibb, and we said we want to do just what you said. And people, you don't have to separate them. Because without getting into a lot of detail, within a couple of weeks, 2 to 3 weeks, you've gotten to that point where PD1's there, and you could do Yervoy first and then the PD1 later, but then you have to get the patient to come back in and all this stuff. So you just what we did was just gave them both at the same time because they last for about 3 weeks or a month. So, you're wasting the first few weeks with the PD1. But
would the same thing happen with the total mutational burden of the tumor? Would if you gave the ipilimumab first, would you then see an increase in the total mutational burden?
No, probably not, because there's nothing inherent that's going to do anything to the tumor except kill them.
So would there be any therapeutic advantage, though, to upping the PD1 PDL1? Would that make nivolumab
that's what we did was we gave the two antibodies together again. We reached this PD1 didn't work because there were no T cells in there. And PD1, we know, cannot drive T cells into a tumor. I mean, not even prostate cancer or kidney cancer. If they're there, it's fine, but it's not going to fix it, whereas CTLA-4 will. And it's a fundamental difference between those two molecules. But anyway, we gave a number of patients that, and there were several. It wasn't randomized or anything, so the numbers don't mean a lot, but there were some complete responses where the patients just, the PSAs dropped, with successive doses of the combination to undetectable. A lot of the metastases went away. Some of these guys are 2 years out now with no disease, and so that led us. But the problem was it was really too toxic, and the doses given were the same ones that had been given in the melanoma trials, combination trials. The problem is, and my wife Pam tried to tell BMS, is that, melanoma strikes people in their 30s and 40s and early 50s, whereas prostate cancer hits old guys, 60s, 70s mostly. And if you've got metastatic disease, you're pretty sick, and they just can't tolerate what the normal thing is. And so we're doing that trial again now, except instead of giving with two variations. One is the IPI's being given 6 weeks instead of 3 weeks apart. And then also in another arm of the trial, they're getting 1 mg per kg instead of 3 mg per kg. And so we're hoping, that we could cut down it, but not so much that,
So even at one, for our audience to explain that's 1 milligram per kilogram of body weight would be like the lowest, not possible, but that would be considered a very low dose, right?
Yeah. And we don't know if it works yet or not, but we'll see.
And you also published a paper on you and I remember that Jed Welchek was also another author on it, of proposing the use of Newcastle disease virus vaccine, or Newcastle disease virus, let's say, as an adjunct to
that was in my lab, actually. We did that. I was going to note, I noticed in your email you mentioned NDV, and I was going to say, I got a fellow that came to my lab from Peter Palazi's lab who had, had developed and attenuated Newcastle disease virus.
Is it identical to the veterinary Newcastle virus, or is it?
I think this one's been attenuated more. It's actually been attenuated so that it's safe even for chickens. It doesn't really cause disease. It's in humans, and so he's been doing this. It's been in humans for years, by itself.
Yes. So it was from this person that you got the idea of using the Newcastle?
Yeah. And so we did it, and it worked. It was an interesting experiment. The way we did it, because the whole idea wasn't just to see if we could get. It's known that Newcastle disease virus by itself, injected into a tumor, has pretty good, oncolytic activity. But of course, what happens if you've got metastatic disease? And so that was what we were at. So we put tumors on opposite sides of a mouse, and then treated one with NDV, and not the other. And so if it was just NDV, we could cure the one that was ejected, didn't have any effect on the other one. Although if we looked at that second one, we could tell that there was a T cell infiltration more than if you did nothing to the other one. But then we asked,
You were injecting the virus directly into the tumor? I see.
and then we also noticed that there was a lot more CTLA-4 on the T cells that were in the tumor after the NDV injection. So we came along and treated them with anti-CTLA-4, and then we could get the distant one rejected as well. And so, and then we did additional work and showed that if you blocked type 1 interferons, you could still cure the one you injected. You could not get the secondary, the untreated tumors to go away. That required both killing the first tumor and alerting the innate immune system, probably by the tumor cells themselves detecting the virus, and then the toll-like receptors signaling that they're infected with virus, and so they start making alpha and beta interferons, which then go and
so would you say this is in the broad category of releasing the brake with the immune checkpoint inhibitors and stepping on the gas with the virus or the immune stimulant? I mean, aren't you in a way combining those two concepts?
It's an in vivo immunization, basically, is the way I look at it. We're vaccinating them, except we're doing it by killing the tumor cells, in situ immunization, is how we call it. Anyway, the Merck picked that up. Merck licensed the idea from us.
Are they going to use just a native Newcastle, or are they going to do a recombinant form?
I imagine they'll do everything. I mean, we've since that first paper, we've been studying for years, both Pam and I, in different ways, the role of a molecule called ICOS, which is induced on cells, T cells. And so if we gave a signal through ICOS by putting ICOS ligand to the Newcastle disease virus. So the idea is it goes in and it doesn't integrate, but it starts replicating in the cells, and in the process of that kills the tumor cell, but it also makes the tumor cells start expressing ICOS ligand on their surface. So when the T cells come in, they get, in addition to prime, they get that other signal, which makes them much, much more effective.
I only have one more question. Could you give me your broad vision of where you think cancer research and treatment will be in 10 years, or alternately where you hope it'll be? In other words, what's your vision? You gave some of that in your Nobel Prize lecture, but I'd like to hear it more explicitly addressed by you.
Well, people ask me all the time, do I think that we're going to do away with cancer? I think some cancers, maybe, with melanoma, I think we're at 60% roughly now. I think if we keep working hard and getting the right combinations, we ought to be able to get that up higher. Other ones, right now we're nowhere with glioblastoma or pancreatic, really. I mean, there's some hints, but I think we're going to start seeing some responses there. But it's going to take a lot of hard work. I think what's going to happen in the laboratory is that essentially every therapeutic regimen that patients receive in 10 years will have an immunotherapy component to it. They may still be radiation or chemo with it. I know some radiation oncologists that have realized, Sandra DeMoria is one, and Sylvia Fomenti at Cornell, who we worked with way back, but they realized that they've been trying to kill the last tumor cell with the radiation. They realized we don't have to do that, because in killing the last tumor cell, we're also damaging the immune system. So what they're doing is studying how to kill enough tumor cells to get this priming, but do it in a way that doesn't kill the inflammatory cells when they come in the thing. And anyway, a lot of people are doing that now. Some of their work is just spectacular. It's at the level now of in lung cancer in patients, irradiating people that have metastatic disease, irradiating one met, and then giving, anti-CTLA-4 and getting systemic immunity. But then what they're doing now is characterizing it, because where the met is important. Some sites are better at getting the systemic immunity than others. And anyway, that's coming along. So, and I think the same thing's going to happen with chemo. You can reduce the amount that you give where it's not as toxic. But I mean, germ cell cancer is cured now with chemo. But, it does recur. But maybe if you added immunotherapy to that chemo, you can get better response rate and prevent recurrence.
So there's a rationale for low-dose chemotherapy as an adjunct to immunotherapy instead of the other way around.
Exactly.
The shoe is on the other foot now.
Yeah, exactly. And I mean, that's the thing about now that it's finally accepted that that immunotherapy has become the fourth pillar. The unique thing about that is that immunotherapy can be combined with the other three, and give them memory, for example, that you get when you get T cells. But the other thing that we've noticed, right, at least recently, is the lesson that we learned from melanoma was, well, the question is, why doesn't ipilimumab work more often than 20%? Well, we know now one of the answers is because there's PD1, and so you put them both together and it's better. So there are another dozen or so. I mean, people argue about the absolute number, but of the checkpoints. And they're, and again, what we realize now that the immune system has to be controlled. And there's probably more attention paid in the mechanics of the immune system to controlling it rather than getting it started.
Well, aren't there also antibodies to tumor necrosis factor that are blocking the activity of TNF in terms of killing cancer?
Yeah, sometimes we have to use them to protect the patient.
Like Enbrel, things like that. But also from the point of view of of course, TNF was too toxic to actually give to people, but you could remove the blocking factors against TNF and use the patient's own innate TNF to attack the cancer, wouldn't you say?
Well, the way that TNF was used for a while anyway was by isolated limb perfusion. Because they would just, somebody had metastases on their legs, you just, isolate that and then pump the TNF through, the blood vessels and stuff in that limb, and then rinse it out. So what we did, we was a clinical fellow in my lab, had this idea. Let's just say I wasn't as supportive as I should have been, but she did the same thing, but with chemo, really nasty. I don't remember what it was, methotrexate or something, very high doses. And, did the limb perfusion with that. And then after they rinsed it out, then gave them immediately IPI. And she had an 85% response rate. And these are patients that had, tumors all over. She was just treating the ones in one limb, one leg or the other, and everywhere they all went away. And it was because, she killed because she just massively killed the tumor cells in that limb and then released it all to go. All the debris goes and primes T cells everywhere in the body, and then lets them loose with CTLA-4. We got some biopsy specimens. There's just sheets of CD4 T cells in the tumors. It's amazing. But anyway, back to 10 years from now, I think the other thing, though, that we're realizing is that some of these other checkpoints, I think that CTLA-4 and PD1 are the big ones. We know CTLA-4 works during the initial priming of a cell, very early in the functional development of a T cell, whereas PD1 works at the end when they're terminally differentiated already and they've been working for a while and they're exhausted, and it just keeps them going for a while. So, and you put them together and obviously, they're really powerful. But in our prostate experiment, for example, the patients that got anti-CTLA-4, we could find this other checkpoint called VISTA. Nobody knows a lot about it yet, except it's discovered by a guy named Randy Noel, and there's some antibodies to it. But So it was also induced in prostate cancer when we blocked CTLA-4. But we looked for it in melanoma, and it was not induced in melanoma when we gave it. So what we're going to have to do is individualize the treatment somewhat by looking at the distribution of these other checkpoints. They're not going to get you from 0 to 50% or something. We're pretty close to 50% now in kidney combinations, but they may help you get from 60 to 70, or, you know, whatever, as long as they don't add toxicity. So, I mean, that's kind of where we are now. You know, it's really being smarter about it. And a lot of biopsies. One of the things that is my pet peeve right now is you can read about there's almost 2,000 trials now of PD1 plus antibodies plus something, and most of that's just, you know, there's no strong rationale for doing them.
It's just empirical. Just throw this together with that.
Our company owns 2 drugs, so let's put our 2 drugs together. But the thing, and that's, I mean, I can't argue say that's useless, but what is irritating, and this is something I learned from Lloyd over the years, and Pam as well, and you know, we talk about it all the time. Lloyd had, I don't know how well you knew him, but he had a lot of little phrases and things that, you know, were usually worth listening to. You know, he just, you know, he said, you know, if you don't learn from every patient, you're doing them a disservice and the field a disservice. And most of those almost 2,000 trials, they just look for a clinical endpoint. They don't get it, and they say that failed and move on. And that's such a waste, because if you got it, you know, you can learn from failures just like you can from successes. And if people would get biopsies from those, because we know what to look for by and large now, but you might be able to see what's, you know, did anything happen that you don't get with a single? You know, and if it was, which direction did it go in? And is there some way you can build on it? You know, and there's so, you know, there's just thousands of patients being wasted because we're not learning.
I agree. Well, look, I took up an hour of your time, and I told your secretary I'd only taken a half hour, so
Okay.
I greatly appreciate this. It's a great pleasure to meet you, honor to meet you, and I hope we can talk again sometime.
Yeah.